Hi, my name is Xeph Ivankovich. My pronouns are they, them, theirs, and I will be presenting on my research on UV mutagenesis and screening of the green microalga Picochlorum soloecismus. First, I want to address why algae and some of the prospects and challenges we face as algal biofuel researchers. Some of the prospects are that it can be converted to a third-generation biofuel, which allow it to be grown on non-arable land and not use freshwater resources. It also has a high productivity in comparison to other land-based biofuel crops. It also can be used as a drop-in fuel when it's converted into a fuel, which allows it to be put into any engine without any sort of modifications needed. It also can be used to make other bioproducts such as bioplastics, as well as nutraceuticals and other nutritional supplements. It also is carbon neutral, which makes it a renewable resource. But some of the challenges we face are increasing the productivity of algal strains, scale up to industrial scale, and the cost of scaling it up to industrial scales. So some background on the strain that I used, Picochlorum soloecismus, is that it is a small 2 to 4 micrometer in diameter green unicellular microalgae. It is able to withstand a wide range of light intensities, temperatures, and salt tolerances. And this is particularly useful because it's able to withstand temperature fluctuations between day and night in an outdoor setting, and also it can withstand differences in salt concentrations, which will happen in an outdoor setting because of evaporation. Finally, it can accumulate over 50% of its weight in carbon storage molecules, which is useful because we use those molecules to create fuel and other bioproducts. So a little bit of background on UV mutagenesis and why I used it. So UV mutagenesis is essentially just exposing cells to UV light. And this exposure is used with short wavelength but UV light. And this short wavelength is more damaging to DNA, which causes more mutation, which is ideal for research. And these mutations that it induces are dimerization of adjacent cytosine and thymine nucleotides. And you can see in the bottom left where the bonds will form to create the dimer. This dimer inhibits DNA translation and replication, and this causes the DNA repair mechanisms to be activated, which cause point mutations, which eventually cause changes in protein structure, which can also influence cell morphology and what kinds of carbon storage molecules it produces. And why it's useful is because it's a form of random mutagenesis, which mimics natural DNA mutation mechanisms, which allow it to not be categorized as a genetically modified organism. And that's useful because we won't have to consult with the EPA on any sort of federal regulations in order to grow it outside. It also helps us to identify genes of interest, which we can then use targeted mutagenesis to manipulate to get products that we desire. So one of the experiments that I did was a large amount of clone screening in 6 well plates. So first we started by UV mutagenizing the algae, and we exposed it to varying amounts of time to UV light, and our ideal death rate was 50% or higher, which was achieved in the 30-second plate. So we took that 30-second plate, washed the cells off of the plate, and grew it in liquid culture. Then we sorted it based on which phase of cell growth the cells were in. So in the replete phase, they're in exponential growth in an abundance of nitrogen. And in the deplete phase, they're in starvation phase, which means they're accumulating carbon storage molecules such as carbohydrates and lipids. We then took 11 replete clones and 12 deplete clones and grew them alongside the parent in 6 well plates in triplicate for a total of 72 wells. I sampled each of these wells each day and took measurements via optical density in order to monitor how much the cells were growing and calculate their exponential growth rates. We also took samples from the 2 days replete cells and the 3 days deplete cells and put them into a flow cytometer in order to measure the cell counts, cell size, and lipid content. So here's some of the data from that experiment. In the upper left, you can see the growth curves that I created using the optical density data. And you can see that compared to the parent, most of the clones did approximately the same or a little better or a little worse than the parent. But once you look at the lipid content, cell counts, and relative size, you can see that there's a high amount of variation compared to the parent in the clones, which confirms our hypothesis that the UV mutagenesis affects DNA and causes mutations. But the clones that we were particularly interested in, though we didn't end up choosing for further analysis, were CR1, CD4, 6, and 8, because they exhibited a phenotype that we later confirmed with microscopy that was a doublet or tetrad phenotype. So that is essentially 2 cells or 4 cells growing together as one. And we were able to make this hypothesis by looking at the fact that it has a much higher lipid content compared to all the other clones, as well as a much higher relative size. They also had higher chlorophyll fluorescence, but that graph is not shown on this slide. So we decided to proceed with a scaled up version of clones that had high lipid content because that was part of our go-no-go goals. So we chose the clones that are circled here for a scaled up version of this experiment. So in the scaled up version of this experiment, we did essentially the same thing, but we grew 5 clones along with the parent in triplicate in 60 milliliters of total volume. Then we took the 2 days replete and the 3 days deplete and put them into the samples and put them into the flow cytometer and measured cell counts, cell size, and lipid content. I also took daily optical density measurements in order to create growth curves. Furthermore, the most important part of this experiment was the fact that we were planning on doing biochemical analysis, which is lipid analysis by gas chromatography and carbohydrate analysis by spectrophotometry, which allows us to see the fatty acids and acid and carbohydrate profile of the cells, which allow us to compare it to the parent and see what kind of changes were experienced by the cells. And here's some of the data from that experiment. And like the last experiment, they grew approximately the same to the parent, but had different relative sizes, cell counts, lipid contents, and even dry weights, though dry weights will have to be remeasured due to their large error bars, especially in CR3 and CD12. But the clone we are particularly interested in is CR2, since it has a higher amount of lipid content and a higher cell count, as well as a higher relative size. And even though CR3 has a higher relative size and lipid content, it has lower cell counts, which means there's a lower accumulation of biomass. In conclusion, exposing the cells to UV light induced favorable morphological and biochemical changes in P. soloecismus, and including increased lipid accumulation on a per-cell basis. We also confirmed our hypothesis that UV mutagenesis caused mutations in the DNA that we saw in our data by DNA sequencing, and we found from DNA sequencing that there was a 49% increase in mutation rate, confirming our hypothesis. Future plans are that we are planning on doing biochemical analysis, including ash-free dry weight, which is a measure of inorganic content, a carbohydrate assay, a FAME analysis, which is a lipid profile analysis, and we plan on continued screening and analyzing the productivity of other selected clones. I'd like to acknowledge the following people on my team for helping me become a better scientist and create outstanding research with LANL. Thank you for watching my presentation.